So this should make you happy. You've made it. This will be the last lecture you'll have to hear from me, at least in lecture, um, over the material. This will be the last part of what is on the lecture exam for exam 5 for the final exam. And so you will not have to hear my voice, at least for the lecture portion, after you're done with this lecture. So we'll be going over the digestive system. We'll look at how materials move through it, how we break down the food material we eat, and then how we absorb it. All right, so we'll go through all those processes. Essentially, we'll walk our way through the digestive tract and all the things that are taking place through the different regions. So digestion and absorption, kind of the main thing. We'll talk about some other functions of the digestive tract, but the main thing is we're going to be doing digestion along the way as we travel down the tract. Right? And we do this two ways. We have our food. Say we're eating an apple, we take a bite of an apple, it's this big chunk of apple. We have to do what's called mechanical breakdown. We have to break it down into little pieces physically. All right, and then we're going to use chemistry. We're going to break it down using hydrolysis reactions. Okay, because what we'll see is three main nutrients. We got our carbohydrates. Okay, what you think of sugars or starches. We got our proteins, and we have our lipids or our fat. And what we're going to see is we have to break these down almost to individual components. You think of carbohydrates, you think of in bread or potatoes, you got starch, and it's this long chain of glucose molecules. Well, we can't absorb this long chain of glucose molecules. We have to get it down to individual glucose molecules. So that is what we're doing for digestion. We're breaking the material down so we can actually absorb it to get absorption. Okay, we'll see that's going to take place in the small intestines. And I say monomers. Monomers are individuals. We're going to see, like with proteins, we can actually break them down to maybe 2 or 3 amino acids, but we have to break them down to these smaller components to get proper absorption. Okay, so we'll talk that process of digestion and that process of absorption as we go along our way through the digestive tract. Let's first look, just kind of a reminder chemistry-wise. If you remember carbohydrates, okay, this would be an individual glucose molecule. When we talk starches, we would have many of these, but they are bonded together for a starch. Okay, starch, we have this long chain of carbohydrates. Okay, glucose, galactose, fructose. Our proteins, we got long chains of amino acids. Okay, you can see our amino acid there. We'd have multiple amino acids making up our proteins, but what we do is we break those bonds eventually. And then for lipids, if you remember, we have triglyceride. Okay. We have this glycerol backbone, and then this would have that long chain of fatty acids. And you see we'll break that down into glycerol and into those fatty acids. Okay, so we'll talk about the process of first breaking down the food and then breaking down, this is the hydrolysis part, where we do the actual, what we call chemical digestion. So when we look at the digestive tract, I think of the digestive tract. The digestive tract is actually considered the outside, even though it runs through it, it's the outside environment. It's lined with epithelial, just like our skin, okay, creating a barrier. So there's a barrier between the food material and actually physically getting into the bloodstream. So you can think of this environment essentially as the outside environment. The food or the material that's running through the digestive tract is not into our actual body until it actually travels through those layers of the digestive tract and gets into the bloodstream and into our body. Okay. We're going to see it's open, both ends, and there's going to be enzymes involved in digestion. We're going to see we're going to be breaking down stuff, and so it's relatively kind of harsh conditions you'll see in our stomach. What do we have? We have a very acidic environment. We also have these enzymes breaking down food, breaking down proteins, breaking down lipids. If you remember, our cells are made up of proteins, carbohydrates, lipids. So it's a very kind of harsh environment. So we want to make sure this material or these enzymes stay and are only active within the digestive tract here. Okay, but what we're going to do, we're going to begin, we'll start talking about our pathway down the digestive tract here. Talk about what's taking place, where we're getting mechanical digestion, where we're getting chemical digestion, and also where we're getting absorption. Okay. Also, to make our way through the path, we got to move the material. So we'll talk about how material and the process of moving the materials through the digestive tract. So some of the functions of the digestive tract we're going to look at. We already discussed that we have to move the food through, so motility. All right, to be able to get to these different regions, we have to be able to move the food through. Okay, as we move the food along the way, we'll see there are some secretions. We have exocrine. We're going to secrete material into the digestive tract to aid in the digestion and movement and so forth. And we're also going to have endocrine secretions by the cells of the digestive tract. This helps regulate movement, helps regulate enzyme release, secretion of other hormones and so forth, and materials like bile that we'll talk about. So as we move through, as we do motility through the digestive tract, along the way, you'll see we are secreting materials in, and also secreting materials into the bloodstream to help regulate the digestive system. Right, the ones we've already discussed, big is breaking down digestion. Okay, breaking down the food, we're going to be able to physically break it down, doing mechanical digestion, and we're going to chemically break it down using those enzymes to break it into smaller components, breaking those bonds in those nutrient molecules. And then finally, we'll get absorption. Absorption, we'll see, most will happen in the blood, but in the case of the lipids, will happen and get absorbed into the lymph fluid. Okay, finally, once we're done with digesting, we're done with absorbing, we still have material left over, some of the waste material, and so we got to store it and finally eliminate it. Okay, get it out of the body. And then along the way, digestive tract has some immune response in that we have an epithelial layer. Remember, this is a barrier. It's kind of like our skin. It's made of epithelium along the way. Okay, so keep stuff out. We only want to bring in the materials, the good stuff that we need for the body, for the cells of the body. And then immune cells along the way. If you remember your little Peyer's patches, there are little patches of immune cells just under the epithelial, and actually some of it's a little bit exposed, in that we are bringing materials through the digestive tract from the outside environment. All right, we have all that good stuff, we have the carbohydrates, we have all those nutrients, but sometimes pathogens can get through there. So it's good that we have these immune cells to start an immune response in case we ingest some of that toxic material, those pathogens or some kind of toxin. Right? So let's do just a brief macro digestion of the division of the digestive system. Okay. We have the main feature of the digestive tract is the gastrointestinal tract, the GI tract. That essentially are the organs of the tube that run from the mouth all the way down to the anus. This is the tract, the tubing that runs through. And we start at the oral cavity, down into the pharynx and the esophagus, which leads us into the stomach, and then we get into the small intestines and the large intestines. Okay, and that'll finally get us to the anal cavity where we eliminate. Right, so that is the GI tract. That is the main part. That is where the food is moving through, or the materials are moving through. But there are other organs that help aid in the digestion. These accessory digestive organs, such as things as the teeth, the tongue, the salivary glands, are going to help in moistening the food and also in some of the chemical digestion. We're going to have the liver. The liver is going to give us some secretions to help break down fats, along with the gallbladder. And then we're going to see the pancreas is one of the main accessory organs in that it provides the means of doing chemical digestion, or a lot of the chemical digestion. Okay, so the GI tract are the organs through the tubing, and then we have the accessory organs usually giving secretions or helping with mechanical digestion that make up the accessory digestive organs. So getting closer into looking at the actual layers of that tubing that's running through the parts of the GI tract, we'll see there are 4 layers, or 4 tunics, we'll say. And the first of those is the mucosa, the inner layer. That's this here. Okay, also this layer here. Okay, or this layer out here on the surface. So this is a layer that's actually in contact with the food material. It's going to be secretory, it's going to have secretions coming out, but it's also going to be absorptive because we have to get in through this layer to actually get into the blood supply. And blood supply, that's in the submucosa, and we have this vascular layer. And also in the submucosa, we have this large nerve plexus. Okay. And this is where we're going to pick up the nutrients, because this is where we have that large vascular layer. Okay, and now just deep to the submucosa is the muscularis. So we're looking now here. Okay, and if you remember from your anatomy days, there are smooth muscle in here. We have actually 2 layers of smooth muscles that are going to aid in helping in the motility and also the churning of materials when we talk about peristalsis and segmentation. Okay, these are movements that are taking place. And these 2 layers of smooth muscle, we have an inner circular layer. So the inner circular layer is basically wrapped around the tubing. So it can constrict it and dilate it, do this kind of movement. And then the outer longitudinal, here's the tubing, the muscle fibers run this way. So this will have us, if we get this kind of dance of the movement of the circular muscle and then the movement of the longitudinal, this will help propel or move material through the nerve or through the digestive system. Okay, and then finally we have the serosus. This is this layer here. Okay, aids in protection, also continuous with the mesenteries. If you remember, the mesenteries help anchor our digestive organs into the abdominal wall. All right, so we'll look there. We'll start, before we get into absorption, we'll look closer into these layers and what's taking place in these cells to help get absorption, and also look at some of the layers and where we see the secretions taking place. Now, nervous system wise, we'll see there is this nervous system. This enteric nervous system is basically this big, like mesh-like network of neurons that basically has local controls the basic movements of the digestive tract. And you see very similar in that, kind of like the heart, where the heart can be regulated by the cells autorhythmic cells. We're going to see there are some cells similar to those autorhythmic cells in the GI tract. Okay, but it also has, you see the black here is enteric neurons, and that they help regulate. They get the sensory mechanoreceptors. We get stretch on the stomach. Hey, I'm full. Chemoreceptors, because we have chemicals running through here. So we get this nervous system in that we get the sensory part, and then we get the somewhat of the motor response. But similar to the heart, the autonomic, the parasympathetic and sympathetic, has some activity over the activities that take place in the digestive tract. You can see the connections here with the parasympathetic running through and the sympathetic. So the parasympathetic, the sympathetic can have some control over or some influence over secretions and also the motility. And if you remember, parasympathetic is rest and digest. So these parasympathetic fibers would stimulate secretions here and would also stimulate the muscle. Here's our muscle layers, help stimulate the muscle layers in contraction, increase motility, where sympathetic would have an inhibitory effect in these areas here. Okay, but this is a large nervous system. A lot of the control of the digestive tract is controlled by the enteric nervous system, and again with influence of the parasympathetic and the sympathetic nervous system. So for motility to take place, we have to contract those smooth muscles layers of the muscularis. And in this case, we have similar cells to the heart, where we have these cells that are depolarizing to cause the contraction of the heart. In this case, we have these cells producing what's called slow waves, producing these depolarizations, and we have gap junctions from these cells into the smooth muscles around the digestive tract, and they're going to help aid in the contraction of that smooth muscle. Okay, they are kind of like the pacemaker activity. Okay, and similar to the heart is we have this autonomic, right? We have the autonomic nervous system that helped regulate, okay, or modify the activity, and the parasympathetic and the sympathetic are going to modify this slow waves. Okay, these are made by the interstitial cells of Cajal. We'll look at the next slide, but they can regulate the activity of these slow waves. In the heart, we saw the sympathetic increase the activity of the autorhythmic cells, where the parasympathetic decreased them. It's going to be opposite here in that the parasympathetic, right, we're in the digestive tract, rest and digest for parasympathetic. The parasympathetic will increase the activity, and the sympathetic would inhibit the activity or inhibit the slow wave movement. And here's kind of the setup, and we have these interstitial cells of Cajal, and it's kind of not shown, but there should be some gap junctions here to show that the slow wave movement of these cells help aid or give us the contraction. All right, and again, the autonomic nervous system influences. Okay, we have the autonomic is going to, or the parasympathetic is going to increase activity here, where the sympathetic is going to inhibit the activity or inhibit the slow wave movement. Okay. This is going to be what helps control the contraction, and what controls the contraction controls the motility that's taking place through the digestive tract. The parasympathetic's on board. We're more active. We're moving stuff through there because we're doing digestion. We don't have to shunt blood out to the muscles when we're exercising. Let's say when we're exercising, now we can shunt the blood to the digestive organ, and we can move stuff through and pick up those nutrients. Now, looking at this motility, we have two kind of forms of contraction that help aid in this motility. We have ones that perform peristalsis, and this is a wave-like. So we have muscles along the way. We have movement contraction taking place here, and then as we contract here, we start contracting down here, and this helps move, do peristalsis, push the material through the digestive system. Okay, this helps in pushing it through. That we can get this kind of alternating modes of contraction of the muscles along the way. So alternating muscle contractions, and this is called segmentation. And this actually kind of moves the material back and forth, back and forth, back and forth. Okay, it's going to aid in what we call mechanical digestion, in that we're mixing up the material and we're further breaking it down to smaller components. So wave-like, peristalsis helps push us through. Segmentation helps churn the material up, make it so it's more of a paste, and we can also help aid in peristalsis in that we can push it through, but it also aids in the actual mechanical digestion of the material with this segmentation. So we kind of have the anatomy down, we got the way of moving it through. Now let's move our way through from the oral cavity to the anus. We're going to move the material through. We're going to go through the different sections. We're going to go through what functions are taking place, what part of digestion, where absorption is taking place as we move through this digestive tract. We'll also look at the accessory organs and where they play a part in this digestion process. Okay, we're going to see accessory organs aid in the ability of the GI tract to function properly. Okay. So as we move, I want to talk digestion. We have two modes of digestion. We have that mechanical, and we have chemical. So as we move through various regions, we'll be doing forms of either mechanical or chemical. Mechanical is that physical breakdown. We got that large chunk of apple, we got to break it down physically into smaller, almost into, say, a paste. When we chew it down into smaller components, that's mechanical. Chemical is where we're actually using enzymes to break the bonds in the molecules. Okay, so mechanical is breaking down the food, but to actually break down the nutrients, break down the carbohydrates, break down the proteins, break down the lipids, we have to do chemical. We have to use enzymes to break the bonds in those materials. So we'll be doing variations of this along the way. Let's start out. We're going to start out in the oral cavity. We put food in our mouth. We start chewing up. Chewing is called mastication, and mastication is form of mechanical, right? We're breaking the food down into these smaller components, the teeth, the accessory organs, the teeth. We also have some secretions there. We have salivary glands that are secreting materials. They're going to secrete mucus, antimicrobial agent, all right? And one of those glands secretes amylase. Amylase is an enzyme. So we're going to actually start some chemical digestion in the oral cavity, and that amylase is going to start breaking down starch, start breaking down the bonds in starch molecules in those carbohydrates. Okay, in the oral cavity, to get down further in the digestive tract, we have to do deglutition, the process of swallowing. And we have basically three components of it. Here we have the oral, pharynx. So we have this movement when we swallow, both components, oral, pharynx, and esophagus. But that is going to finally bring us down into the first big organ of the digestive tract, and that we move into the stomach. Stomach is big, where we can store food. All right, we're going to initiate digestion, so we're going to initiate chemical digestion of proteins here, and that the enzyme pepsin is released. What else is released here is we release acid. We have this extremely low pH environment. Okay, it allows us to kill bacteria, help aid in activating some of the enzymes. So this pH is important. We need to maintain this pH in the stomach, or we're not going to get proper digestion. But we don't want too much, because remember, you can get acid reflux, and then you get corrosive. It can be corrosive going in the wrong regions. Also, the stomach. Remember, we saw the 2 layers in the stomach. We actually add a 3rd layer in the muscularis and an oblique layer. And so this aids in that we can churn food up very well. We can do those muscle movements, and we can churn the food up, and we churn it almost into this paste-like material called chyme. Okay, so that's going to aid in that, again, mechanical digestion. We're breaking it down. And why do we have to do this mechanical digestion is we got to have those enzymes. All right, if we had a big chunk of food, the enzymes can work on the outer layers, but it couldn't break down the stuff in the middle. So we got to get this food broken down into this small components so the enzymes can actually have activity on all the material, or as much as possible. Okay, what we're doing in the stomach, let's look at closer into these functions that are taking place. Okay, if you remember the Rugae folds, the Rugae folds allow, because one of the things, we store food, allows distension, allows us to expand the stomach. We'll also see in these folds there are the gastric pits, where we're going to get secretions of materials. Okay, because those pits are lined with several cells that are going to secrete different secretions. Also, just looking here is those layers of the muscle. We have the circular layer, we have the longitudinal, but we also have this 3rd oblique layer. Again, that's going to allow us that churning ability in the stomach. But let's look at these gastric pits and what are coming from them. Okay, we have mucosa. So here's the pits coming down here. Okay, they are lined with cells that aid in various secretions. Okay, we got the mucosa neck cells. What do you think they do? They secrete mucus. We're going to help. Remember, this environment is we're making this a very low pH environment, at least in the stomach here. So we need to have that kind of mucus coating, because that pH, that low pH, can be corrosive to the cells. So we got to have kind of a layer of mucus to help keep those cells that line the stomach, keep them from getting corrosive, help protect them. Okay, the parietal cells. Okay, these bigger red ones, they are going to secrete. This is where we get the low pH environment, because they secrete the hydrochloric acid. They're the ones that are going to maintain the low pH in the environment, and we're going to see that helps aiding in ridding ourselves or destroying some of the bacteria, and it also is going to aid in activating the pepsin. Remember, pepsin is an enzyme that is going to help break down proteins. And what we see, they're also called zymogenic cells, and that pepsinogen is actually what is released by these cells. It's a zymogen. Zymogen meaning it is inactive. It's an inactive enzyme. So pepsinogen is inactive. And why might we want to do this? Okay, because remember, the active molecule, pepsin, breaks down proteins. It cleaves bonds in protein molecules. What's in our cells? Yeah, we have lots of proteins. Proteins are the functional molecules in the cells. So would we want pepsin running around being active inside the cell? It'll start chopping up the proteins inside the cell. So we don't want it active. We only want pepsin active when it is secreted out into the digestive tract. So what these cheap cells is, they make pepsinogen. It's inactive. It is a precursor to pepsin. All right, and when released here, we'll see the pepsinogen gets activated to pepsin. And that is one of the functions of that hydrochloric acid. That low pH allows us, we'll see, to turn pepsin into pepsinogen, cleave it into pepsin to make the active, because we do want pepsin active here, but not active in the cell. Okay. And then finally we have chromophene, secrete histamine and serotonin, help regulate some of the activities of the digestive tract here. And also the intrachromophene secretes gastrin. We're going to see one of the hormones that helps regulate the release of hydrochloric acid from the chief cells. So here we'll see the process of hydrochloric secretion. So what do we need for hydrochloric acid? We need a proton and we need chloride. All right. And we're going to see how this makes place. We'll see proton pumps actually produce the hydrochloric acid. They produce the pump going out. Chloride moving out through facilitated diffusion. But how do we get here? How do we get these protons made is we're taking a carbohydrate and water, CAs, that carbonic anhydrase, and we're converting these into carbonic acid. Right? If we make lots of carbonic acid, we're going to produce proton and bicarbonate. And what we're going to see is with this is the blood here, this is the lumen here, we're going to swap out bicarbonate for chloride. All right, and so now in the cell, what do we have? We have our chloride and we have our proton. And if we have those we'll see the protons get pumped out through primary active transport. So we use ATP to pump out the proton, and because there's high chloride here, from bringing it in here, we can do facilitated diffusion to move out the chloride. So we get our hydrochloric acid into these cells. All right. What happens when we start throwing up? You can see we're making this, but as we make the proton, we're also making bicarbonate and putting it into the blood. So what would happen if we started throwing up? We start throwing up, we're losing our protons. Our protons are going with, okay, as we throw up, we're losing it in the stomach. So we have to replenish it. So what do you think that would do to the blood, pH-wise? Right? Typically we do this in a certain amount to maintain this low pH, but now we're losing this, and so we have to constantly be making these protons, but as we make these protons, we're putting bicarbonate in the blood. So what might that do to the pH? Yeah, it would raise the pH because I would be putting in more bicarbonate into the bloodstream because I have to replenish these protons that typically were already out there. Now I'm losing them through the throw up. That's going to cause this to occur more, and I get more of this in the blood, and therefore my pH in my blood is going to go up. Okay? So you see the relation between the secretions in the digestive tract and that, and what affects the pH in the blood. We already discussed some of the role of that low pH, but that low pH makes the environment, makes it harsh for any microorganisms. Also, the pH can help denature the proteins or proteins need to be unraveled. We need to have access for the enzymes to get into those protein molecules. But also, we already discussed how acidic conditions can help activate pepsin. Okay, pepsinogen. There's pepsin out here. The hydrochloric acid activates the pepsin, and pepsinogen can then get cleaved into pepsin. It's almost like a positive feedback in that the hydrochloric acid, pepsin, helps cleave pepsinogen to pepsin, and then more pepsin will cleave more pepsinogen. Okay, but this takes place because this hydrochloric acid helps activate that pepsin. If you remember our pepsin molecules, its activity is highest in low pH environments. Okay, whereas enzymes, where their activity is good in neutral environments, they would not be active here. But pepsin is active in a low acidic or low pH environment, and therefore we'll get higher activity of pepsin in the stomach. Now, after the stomach, we're going to move the materials into the small intestines. And the small intestines divided into 2 or 3 components: the duodenum, the jejunum, and the ileum. The small intestine is a long stretch of the digestive tract. If you pulled it out and unraveled it, it would be about a little bit over 20 feet in length. So a very long stretch of this digestive tract. It just wraps back and forth in your abdominal cavity. But the first stretch, the duodenum, right? This is where we're going to get secretions from those accessory organs, the pancreas, the liver, the gallbladder. They're going to aid in chemical digestion of the fats, of the lipids, of the proteins, and of the carbohydrates. So we get secretions, and we'll talk about these secretions in a bit when we look at the liver and the gallbladder, and also the pancreas. The pancreas is going to aid in most of the enzymes that will take place that do the chemical digestion of those 3 nutrients. So here's where we're going to begin. The bulk of the chemical digestion is going to begin in the duodenum, because this is where we're getting the pancreatic juice. Right? And after the duodenum, it is a small stretch, very small stretch, which basically wraps around the stomach there, of the small intestines. After the duodenum, we move into the jejunum. We're going to finish chemical digestion here, because remember, we got to break those molecules down into either 1, 2, or 3 molecule chains. Okay, we can't absorb those big chains of starches, of proteins. We got to break them down into those small individual components. Right, so we're going to finish the digestion, and this is where we're going to get primary region where we're actually going to get absorption of those nutrients into the bloodstream. The ileum, we're going to finish up absorption, and then we're going to pass into the large intestines. Okay, but let's first look at the histology of the small intestines. If we cut into it, you'll see these kind of ridges as you move through here. Those are those circular folds. Or remember from your anatomy days. The circular folds here. And then if we took a snapshot of that, you would see we have these villi on here. You can kind of see the villi here. And then on those villi we have these microvilli. So you see we have these fold upon fold upon fold. We have these basically like 3 layers of where we're getting this folding, and that greatly increases our surface area. Okay, what are we trying to do? We're trying to do absorption, so we want this large surface area so we can move materials in through it. So that's why we have this fold upon fold upon fold, to give us this large surface area so we can absorb things more readily. Okay, but first is these larger circular folds, then the villi, and then the microvilli. This villi here is this, what you see like right here. Okay, so fold upon fold upon fold. So we get this large surface area. And one thing to note too is if you remember the green, the lacteals, we have blood supplies coming in here. So remember, what are we trying to do? We're trying to take the nutrients from lumen and bring it into the body, we're going to see into the bloodstream. As for the lipids, we're actually going to bring it into the lymphatics. And if you remember, these green structures rolling up here are the lacteals of the lymphatic system. Also along, because this is where we're finishing up the digestion. Okay, we have the enzymes from the pancreas. It starts breaking down the proteins into smaller units. And once we get down to into the smaller units, we have another set of enzymes that are on the plasma membrane of the small intestines. Okay, these are called the brush border enzymes or interstitial enzymes. Okay, they are actually proteins that are embedded, and their active site faces the lumen. And they're going to take the molecules, they're going to finish up the digestion. As we've gone down, we're slowly been breaking down into smaller and smaller components, but we got disaccharides. Okay, we're going to see for carbohydrates. Carbohydrates, we have to break the carbohydrates down to the individual molecule. All right, glucose, fructose, or galactose. We can't have a disaccharide. So we have these brush border enzymes that help aid in this, or help finish this digestion process. We got sucrase, maltase, lactase. Many of you have probably heard of lactose intolerant. People that can't eat dairy because dairy is high in lactose. And what they're lacking is they have a non-functional copy of lactase. So they're not able to break down lactose into glucose and galactose, so that lactose stays throughout their digestive system and causes that bloating and kind of discomfort. Okay. For amino acids, we got the peptidase, aminopeptidase, and interkinase. These are going to break down in the middle. They're going to help break down the peptides. We're going to see for peptides, to be able to absorb, we can have them into 2 or 3 amino acids. We can absorb those tripeptides. Okay, but we can't have these long chains of peptides that can be absorbed. So we got to get down to 1, 2, 3 peptides is the max in which we can actually absorb. Okay, in these enzymes, we have brush border enzymes that help get us down to those smaller components. Okay, so let's look in. After we've gone through the small intestines, we've done most of our absorptions. Okay, we've done most of our absorption of our nutrients, of our carbohydrates, our amino acids, and our lipids. Okay, and the large intestines, okay, they are much bigger in size, but they are shorter in distance. We're going to get a lot of absorption of fluids and ions here. And ultimately, we're coming to the end of the chain, so we're going to start taking whatever's left over, the waste, and compact it into feces. Okay. Many of you have probably heard of probiotics. Here is where a lot of our probiotics live, our intestinal flora. Okay, we have many bacteria, good bacteria that help produce vitamins, facilitate this, absorption, and help digest the plant matter. Plant matter has that cellulose. Many of this is where we get, these are our bacteria, help us create our gas also. Okay, but that's what's taking place in the large intestines. We're finally coming to the end of the chain, compacting in the feces, because we're going to eliminate what is left over. All right, so after we've gone through, passed to the rectum, right, it's stored in the rectum, and then as rectal pressure increases, similar to what we see in the urinary bladder, we get the reflex of defecation, and hopefully, again, we're close to the toilet, so we can make it to the toilet. All right. Now that's there. We're going to jump back. We're going to look at kind of absorption, we're going to look at some of the accessory organs, and then we're going to actually look at the absorption process that takes place. All right, so first of the accessory organs, we're looking at the liver. And the liver, and besides, we'll see much of our nutrients are going to go to the liver when they come straight into the bloodstream. They don't go straight into the general circulation. The carbohydrates, amino acids, are going to go to the liver. Okay, the liver also produces bile. The cells produce bile, and this is going to be important in the breakdown of lipids. Okay, if you think of lipids, they're in an aqueous environment. What do lipids want to do? They'll glom together. Okay, if they're in this ball together, there's lipids in the middle, lipids on the outside, and remember our chemical digestion. Our chemical digestion needs to have access to those molecules to break the bonds. All right, if our lipids are all balled up together, there's no access to there, and the enzymes can only break down the outer portion. Right, what the bile does is help emulsify, helps spread out the lipids, so now we can get the enzymes in to break down the lipases. Okay. Another function of the liver, detoxify. Probably those of you that like to go out to the bars on the weekends, you are greatly using your liver because they are where we go to detoxify that alcohol. Interconversion of nutrients, you've seen a bit of the liver in making gluconeogenesis, making new sugars. It is where we get this conversion, makes the ketones. Okay, so it's highly involved in our metabolism. Livers can store nutrients, as in glycogen. See, make blood proteins and perform some phagocytosis of, so red blood cells go to the liver and the spleen to get phagocytosis to break down. Okay, so it's a pretty important structure in all of the functions in metabolism and nutrient and so forth. So definitely take care of your liver because it performs a lot of functions for us. So one thing, we'll talk gallbladder. Gallbladder is involved with this portion of the liver. The liver produces the bile, where if it's constantly producing bile, if we don't need the bile, if we're not eating, a big triple stack at In-N-Out, we don't need to break down all these fats, the bile will get shunted off to the gallbladder to be stored, and then when we eat these fatty foods, we can secrete the bile. The bile's ready to go, and we can emulsify and break down those fats that are in that food we're eating. Right. So, as we said, the liver is going to receive the nutrients. Okay, and this is where we have that portal system. Portal system, usually we have artery, one capillary bed, and then this should be going off to the vein. For most of our tissue, that's how it is. There's a vein here. All right, but in a portal system, we have two capillary beds. We have one capillary bed. In this case, it's connected by the hepatic portal vein that sends it to another capillary bed before we get out into the general bloodstream here. And where we have the capillary beds for the hepatic portal system is the first capillary beds are the capillaries that are wrapped around in the small intestines. When we absorb the nutrients, they are going into this capillary bed. That nutrient-rich blood is going to be sent via the hepatic portal vein to another capillary bed that go to the cells, the hepatocytes of the liver. And the livers are going to basically process the nutrients, and they're seen through there before the blood is actually sent out into the general or systemic circulation and out to the heart to be spread around. Right. So here's the liver anatomy. We got, if you look at it, it has these hexagon-shaped lobules. It's kind of cool looking in these lobules, and you can see broken down there are these structures here. These little cream structures are the hepatocytes. Okay, and you can see branches of hepatic duct, the green. Okay, hepatic portal veins, the purple or bluish coming in. That's bringing the nutrients in from this direction, and the nutrients head this direction. And then the hepatic artery. Okay, the hepatic artery functions to bring in oxygen-rich blood. That's coming from the systemic circulation. It's bringing the oxygen-rich blood because, just like any cells, the hepatocytes need oxygen to make ATP to perform their function. The portal vein is bringing in that nutrient-rich blood. It's coming from the that capillary beds that wrap the digestive system and picked up the nutrients. That's what it's bringing in. The blood flows in this direction towards the central vein. Okay, but as the blood flows through, oxygen gets picked up and the nutrients get picked up and get processed by these hepatocytes. In the other direction, the hepatocytes are making bile and sending it into those little, if you remember, caniculi that feed into these hepatic ducts that ultimately are going to join together and feed into either go all the way to the pancreas, or sorry, all the way to the duodenum, or the bile is going to be shunted to the gallbladder. Okay. So you can see, looking a little bit closer, the lobules, we got the hepatic cords are the cords of these cells, and you can see they line those ducts. They line where the blood, you see the blood actually merges together. The oxygen-rich with the nutrient-rich merges here as it heads towards the central vein. And then the little green caniculi, these here, are where the bile that's produced by the hepatocytes is brought in, and then the bile is going to be brought down to ultimately the common bile duct. The other cells, hard to see here, but these little cells that line the blood that's coming in from the digestive tract, because what else is taking place in the digestive tract? We may actually pick up some pathogens, right? Some either bacteria or viruses, and that is what these Kapuffer cells are there. They're part of the immune system, and they can phagocytose any of those pathogens that they find. So that blood that's coming in gets filtered past these immune cells. So here's coming down. If you remember from your anatomy days, we got the ducts coming in, feed in, come to the common bile duct, or sorry, common hepatic duct, right? We have a left and right hepatic duct merge to make the common hepatoduct, and then it goes here, and this can either go towards the duodenum if we need it right away, or if we're not using the bile that's being produced, it can be shunted to the gallbladder. And then when we eat a fatty meal, the smooth muscle of the gallbladder is basically stimulated by this cholecystokinin, CCK hormone that will cause the smooth muscles to contract, and then the bile will be pushed out and goes into common bile duct. Okay, off to the duodenum, right? Because the bile is going to be used to emulsify the fats, so we can get a proper digestion and absorption of those fats at the small intestines. All right, so there it is. Okay, there's the roll. Here's the left and right merge to make the common hepatic duct. Right, that merged with the cystic duct here, And so what happens is, in bile, if we're not eating a fatty food, it can be brought up into the gallbladder. Then when we eat a fatty food, that stimulates the muscles, smooth muscles to contract here and causes the bile to be brought out through the common bile duct to the duodenum. Right? And that's where it's going to start to aid in the digestion of the fats, help emulsify the fats so the enzymes can get in and break down the lipases, those enzymes that break down the fats. Right? A big player in our chemical digestion is going to be a pancreas. We've seen the pancreas in the endocrine function. Only about 1% of the cells of the pancreas are there to make that glucagon and make the insulin. They are part of that endocrine function. Most of the pancreas is involved in exocrine function and involved in producing the enzymes for digestion. And you see the cells throughout here will be producing the different enzymes and secreting them into this duct. Ultimately, they reach the main pancreatic duct, which will lead into the duodenum. Okay, it produces what's called the pancreatic juices. Okay, combination of enzymes. We're going to see a combination of bicarbonate. So remember, what is coming in here is that highly acidic, right? We're coming from the stomach here into the duodenum. It's very acidic. We want to neutralize that. So the pancreas creates or produces bicarbonate that it makes and sends in part of the pancreatic juice to help neutralize that acid. Okay, because that acid is corrosive. We don't want it running through our whole system. In the stomach, we got ways to deal with it, but if acid's making its way, we don't get neutralized, it's going to be corrosive through the small intestines there. All right, so here is kind of the setup of these in the pancreas here. All right, we have acinar cells. That's the bulk of the cells here. And what are they going to be making? They're going to be making the enzymes. They're going to be making hopefully about 22 kinds of enzymes, different enzymes that break down carbohydrates, that break down the proteins, lipases that break down the lipids. Okay, and they are stored as zymogens. If you remember zymogens, they are inactive. They're an inactive form of that enzyme because we don't want enzymes that break down peptides or break down carbohydrates or break down lipids to be active in the cell because it will be destructive to them. So they're zymogens, they're inactive. Once they get into the lumen and get secreted, that is where they get activated. Okay, the ET cells. There's ET cells for that small pancreatic ducts that produce the bicarbonate. They will produce the fluid that is going to neutralize that acid. So we get these enzymes and this bicarbonate, part of the pancreatic juice that goes into the duodenum. We're going to start neutralizing the acid, and then those pancreatic enzymes will start doing that chemical, the bulk of that chemical digestion, breaking down those bonds and those nutrient molecules. Okay. As we said, the juice has enzymes, trypsins, to break down protein, and contains many other. We have 22 different enzymes. So we got enzymes doing various things on protein digestions, various enzymes doing lipid digestion, and various enzymes doing starch digestion. Okay, but we got trypsins, lipase, amylases that are going to be involved in breaking down these molecules. And then also we have the bicarbonate. We have that buffer to neutralize the acidic material coming from the stomach. Let's look at this secretion of bicarbonate. kind of the opposite of what we see in the stomach. In the stomach, we made acid. Okay, and we made acid. And what happened if we threw up? We threw up all that acid. We had to replenish it. It actually made our blood more basic. Let's see what happens in this case when we secrete bicarbonate. So here we go. Where are we taking? We're taking CO2 from the blood, okay, transporting it in, or we can also transport in bicarbonate, okay, directly from the blood, okay, using secondary active transport with sodium. Okay, so we can get bicarbonate this way, just taking it from the blood, or we can take CO2 from the blood, and what do we have? Carbonic anhydrase, convert it to carbonic acid, and what do? We make bicarbonate, but we also have the proton that will be secreted back into the blood. Okay, and then we get a shift of bicarbonate with chloride because of the charge difference, and there we get, now we get our bicarbonate into the lumen. Okay, that is how we're going to make bicarbonate. Now, when we have diarrhea, we actually lose this very readily. We lose the bicarbonate. We're sending stuff through, so we gotta, what do we have to do? We have to replenish. We gotta keep this environment and being able to neutralize the acid, so we have to increase this activity. If we increase this, what do you see happening? We're pulling out CO2, making this bicarbonate, and what are we creating? We'll be pumping in more protons and pulling more bicarbonate out of the blood. So in case of diarrhea, what we saw with throw up, it was we lost the acid from the stomach juices and it made our blood more basic. Well, what do you think would happen in this case if we had diarrhea and we were losing that bicarbonate? Yeah, we would start making our blood more acidic, making more protons here and pulling bicarbonate out because we have to replenish that bicarbonate that we're losing from the diarrhea. All right, so important stuff to get. You want to make sure you keep your fluid intake and so forth, make sure everything's okay when you either throwing up or having that diarrhea, because it will affect a bit of your pH in your blood. Yeah, you can see pancreatic juices. Here is most of the enzymes. What do we say? They're released at zymogens. We actually have brush border enzymes that help cleave those zymogens into the active enzyme. Okay, trypsin breaks down peptides. It can cleave peptides. We have enzymes that, brush border enzymes that cleave peptides, and so the pancreatic juice, trypsinogen is the zymogen. It's not active. It gets broken down by this brush border into trypsin. Now trypsin is active. Trypsin also does what? It cleaves proteins. So now trypsin can cleave some of those other zymogens, those other enzymes that are made for breaking down lipids, that are breaking down carbohydrates. That trypsin can now activate those enzymes. Okay, so this is how we get activation, because again, we don't want to have active enzymes in our cells. Otherwise, we're going to be causing havoc in our cells and breaking things down that we don't want broken down. Right? So now let's look at a couple of the hormones. I just want you to look at cholecystokinin, okay, and gastrin are two of the major ones that are players in the GI hormones. Okay, gastrin gets secreted. It's going to activate those cells, the chief cells, and the cells that release pepsinogen. Okay, so if you eat food, you're going to start getting secretions. That's going to stimulate gastrin to secrete, and it's going to stimulate the parietal cells to secrete HCl, and the chief cells secrete the pepsinogen. And then we've already touched, we saw a little bit of cholecystokinin. Cholecystokinin is going to be involved chiefly in when we eat fatty foods. It's going to contract the gallbladder. It's going to stimulate the pancreas and motility and so forth to help maintain and move the materials through the digestive tract. But one of the key cues is a fatty meal going to be stimulated because we're going to need to contract that gallbladder to get that bile out into the system. See, there are more, but these are kind of the two main players hormone-wise regulating this GI tract. So we fully looked at the process of digestion and how we get down to these individual molecules. Now let's look at actual absorption. So for carbohydrates, we're going to have to get the molecules down to the monomers, to the individual molecules. So in that case, we get them down to glucose, galactose, or fructose. Okay. And you can see the movement. You can see again our sodium, our glucose and galactose, SGLT1, sodium glucose transporter 1. And what are we doing? We're doing secondary active transport. We're moving glucose and galactose in via the concentration gradient of sodium as energy. Fructose is very low in the cell compared to the, so there we can use facilitated diffusion through the GLUT5 and then moves through via the GLUT5 here. And I didn't say, but glucose and galactose, as those levels build up higher than what's in the blood, they will move through facilitated diffusion into the blood. Okay, but again, carbohydrates, we got to be down to the individual monomers. All right. Peptides, proteins. All right, there's our chains. We get those trypsins out from the pancreatic juice, starts breaking down these very large peptide molecules into smaller and smaller units. Ultimately, we need to get the peptides down to either 1, 2, or 3 di or tripeptides. Okay. Those are what can be absorbed. And similar to what we saw with the glucose and galactose, it'll typically move down using a co-transporter with sodium to move across the membrane here. But you also see even the dipeptides and tripeptides that move into the cell, they will be further broken down into individual amino acids before they are brought or absorbed into the bloodstream. Okay, and again, most of the transportation takes place via sodium and using a sodium gradient. Okay, but different from the carbohydrates is we can absorb, at least from the lumen, we can absorb mono, di, or tripeptides. But when we get into the blood, it's going to be in individual amino acids. Okay. And the more complex of the bunch is the absorption of lipids. We've seen the emulsification by bile, and then the lipids are broken down. Lipase is the enzyme that's going to break down, and it breaks down the triglycerides into 2 fatty acids and 1 fatty acid combined to glycerol, so called a monoglyceride. Okay, so this is where we're at here. When we break down to this, then what happens is fatty acids, they're packaged into these micelles, little balls of fatty acids and monoglycerides, and this is what is absorbed in through the microvilli. Okay, as we enter the cell, the fatty acids and monoglycerides are converted back to the triglycerides, and then they're going to aggregate with cholesterol and proteins and phospholipids to form what's called these chylomicrons. That's what you see here. And the chylomicrons is what's going to be absorbed first to the blood and then into, remember, the lipids are going to be absorbed into the lacteals, the lymphatic tissue. So they don't go directly to the liver like the carbohydrates and the proteins or the amino acids. They go to the liver for processing. These will go into the lymphatic and then into the bloodstream. Okay, but a little more complex in that we first come in as micelles. Basically, my cells are built from this. My cells are converted back to this form, and then we aggregate with cholesterol, proteins, and phospholipids, and the chylomicrons is what gets absorbed into the lymph. So since lipids are made of lipids and they're hydrophobic and they're passing through the blood, they are transporting these chylomicrons, and as they transport it, they acquire these apolipoproteins. Okay, and there are receptors or binding sites at various tissues in which they can bind to these lipoprotein lipase here, and then those lipases will free the fatty acids to be able to enter the tissue. Okay, there is a remnant particle containing cholesterol that will remain in the blood. Since we're looking at lipid transport, let's look at some of the lipoproteins you probably have heard of in health news and so forth. Your liver is going to produce cholesterol and triglycerides and send them in with apolipoproteins, and they make these very low density lipoproteins that can transport triglycerides to the different organs throughout the body. After the triglycerides are removed, we're left with low density lipoproteins, and these low density lipoproteins can transport cholesterol to the various organs, and if high levels, they'll transport some to the blood vessels. That's where you get the LDLs that cause those atherosclerosis, those plaques, giving you heart disease, right? Because if these build up and cut off blood supply, you're putting enough blood supply to those organs. So in the news, you've heard these LDLs, bad cholesterols, since they deposit these plaques, where the high density lipoproteins are known as the good lipoproteins or cholesterol, because they'll sop up the extra cholesterol, remove the cholesterol, bring it back to the liver, and the liver can now process that cholesterol. Okay, so LDL is good, HDL is bad. Common look if you're taking blood work is to look at LDLs over HDLs. Even if LDLs are a little bit high and HDLs are high, then kind of offsets it. But if you have high LDLs and low HDLs, then you're starting to look at some more risk at getting this heart disease and getting these plaques. And so that will be it. This is the last slide of the semester 4 lecture. Hope you guys had a decent semester at least. I know it's hard learning this stuff online, but it's been a pleasure teaching you guys. But study hard. I hope you guys all do good on the last exam and give you all good grades. All right, take care.